Welcome back to day 2 of Advances in Proteomics and Metabolomics. We're excited to have you here, and we can't wait to see the incredible insights and connections that will emerge today. My name is Princess Sennett, conference producer at Technology Networks, and I am pleased to welcome our opening talk today, brought to you by Dr. Aivett Bilbao. Deciphering Microbial Omics Signatures in Multidimensional Mass Spectrometry. Dr. Bilbao is a computational scientist in the Computing, Analytics, and Modeling Science area at Pacific Northwest National Laboratory. Dr. Bilbao's work is at the forefront of cutting-edge research, where she delves into the world of computational tools for mass spectrometry-based omics. She earned her PhD from the University of Geneva in Switzerland, with special interest in data-independent acquisition mass spectrometry. Dr. Bilbao possesses a unique dual expertise, excelling in both algorithm design and software implementation. This enables her to effectively process data from a variety of mass analyzers, including time-of-flight, quadruple, and Fourier transform-based instruments. Her contributions continue to push the boundaries of what's possible in the world of omics. Following the presentation, we will have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. Now, without further ado, I will now hand over to Dr. Bilbao to begin her presentation.
Thank you, Princess, for the introduction. Welcome, everyone. It's my pleasure to be here. Thank you for this invitation. I will now talk about our recent work. In this outline, I would like to give you an overview of what I will present today. I will start with a brief introduction about ion mobility, spectrometry, and data-independent acquisition mass spectrometry. Then I will explain how we combine these techniques with mass spectrometry and also liquid chromatography to perform multidimensional measurements. Next, I will describe our new algorithm called PeakDecoder that I developed and we use in these two projects using these multidimensional measurements for synthetic biology and plant matter degradation. And finally, I will talk about our current progress and work developing PeakDecoder for omics measurements, including also proteomics. Ion mobility spectrometry is an analytical technique that provides rapid gas phase separation. It's increasingly being coupled with mass spectrometry to improve omics measurements, and because ion mobility mass spectrometry can distinguish isomers and isobars, which are impossible to distinguish by mass spectrometry alone. And using ion mobility, we can compute or determine measure collision cross-section, which is a robust physical-chemical property reflecting the size and the shape of ions, and we can use this to increase our confidence in molecular identification. In this example, we can see how using the collision cross-section, again, this property is related to the size and the shape of the molecules. We can see that as smaller molecules will have a smaller collision cross-section, and then different structures, even if they have the same mass, can be separated by this analytical technique. And we can see that looking at also at trends, patterns of these collision cross-sections, we can distinguish different kind of molecules. For example, biomolecules, we can separate them compared to halogen and other type of molecules that we can find in different kind of samples. Now, in these multidimensional measurements, how we combine ion mobility with liquid chromatography is because of the time scale, the compatibility, we can insert the ion mobility separation between the liquid chromatography and the mass spectrometry. And then this means that rather than having a single mass spectrum at every time point or every chromatography point, we will have what we call a frame or an ion mobility separation. Then we can separate further between the LC and the mass spectrometry, the ions based on the size and the shape of the molecules. Now, regarding fragmentation, how we can collect fragmentation data. As you may be aware, we have there are two main techniques to collect fragmentation data. The traditional one, it's a data-dependent acquisition, where the instrument is set up to select individual ions and serially isolate them and fragment it to collect the MS2 or MS spectra. So the difference compared with data-independent acquisition is that it systematically fragments all the ions within a given time point without doing selection of single ions. And this allows us to collect data that has no bias against the low abundance ions, means it will fragment all ions that are eluting at that time point. And we can build chromatograms also for the fragment ions. That's the main difference between DIA and DDA. And the way how we combine then this fragmentation technique, in this case DIA, with the ion mobility system, we use the ILN6560 drift tube, specifically ion mobility type. We can operate the instrument to alternate between what is called low and high collision energy. It means we can collect one at every LC time point. We can collect one ion mobility separation that has no collision energy, so we can collect spectra at the intact mass or the intact precursor signal, and then we can activate the next mobility separation with collision energy, high collision energy, and then since this happens after the chromatography and the mobility cell, we can collect fragmentation spectra that will align in time both retention time and mobility with the precursor. This is a very rich data, but the limitation is that it's very difficult to process and the traditional tools don't work. So we developed this new algorithm that we call PeakDecoder that I designed, and to enable the processing, the annotation for this kind of complex data. We combined a series of existing tools, also together into a workflow that includes includes some machine learning with a new code that we developed into a single workflow. So the first stage is the training in our algorithm. For this step, we use MS-dial to do untargeted feature detection. So for this kind of data, DIA kind of data, and with ion mobility, it can be processed either in untargeted or targeted mode. Untargeted means that we detect all the peaks, and then we extract the fragments that align in time with the precursor, regardless of their annotation or the identity of the metabolites. So the way PeakDecoder works is we detect those signals first using MS-dial, is performing the fragment ion detection and the deconvolution, the precursor and the fragment deconvolution from the raw data itself. Then we extract these signals and we employ our code to generate decoys. From those, we take the high quality P groups, we call is the precursor and the aligned fragments, to generate decoys, targets in decoys. And then we use Skyline to extract them in a targeted fashion. It means we know the mass to charge and the retention time of the precursor and fragments. And then we use Skyline, another very popular tool, to extract these signals in a targeted way. There are different advantages for each of these modes of processing that you can read in our paper. It's discussed here in the reference. Using Skyline, we can extract then these metrics from each of the P groups that we can use to evaluate if it's the quality of the signals, if they coelute and have good comobility. Then finally, since we have these signals, where we have the targets and the decoys. The targets are the high quality P groups. The decoys are actually fake signals that are being built that they don't have good coelution. So in this way, we can train a model, a binary classification model. And in this case, we use a support vector machine where we can then tell it, okay, learn using these metrics, this is how it looks for good when the coelution and the comobility is good quality, and this is are the metrics how they look when it's not good, and that's the basis for us to detect if the molecule is present or not in the sample. So once we have this mole train, then we have our library that we built. We created this library by analyzing standards where we have the retention time, the collision cross sections, and the masses of the precursors and the fragments. We again can use Skyline to extract these peak metrics for each of the molecules. And then we use this model that is already trained to score these signals, and finally we can do filtering, and we have a confidence score that tells us if the molecule is present or not, and we can do quantitation also based on the extracted peak areas. In the first step, we do the deconvolution. As I mentioned, this is untargeted feature detection. It's independent of the identity of the molecule. It's just detecting the precursors, MS dial detecting the precursors and detecting, deconvolving the fragments that has the same time and profile, mobility and retention time profile as the precursor. Then we take the high quality ones of these deconvoluted peaks or peak groups. And then we perform the decoy generation. For the decoy generation, basically, we sort this list of high quality precursors to pair two of the targets with using different criteria. For example, they have to be at least 3 minutes far away in retention time, so we know it's not the same molecule, and in similar mass, not a too big mass, so we can increase the probability that it's a similar molecule. We are not generating decoys that are not really, they are invisible in nature. So for each pair of targets that are grouped together, then we generate decoys, 2 decoys. And what we do is we swap fragments. We keep the same precursor, swap the fragments, and then in this way, when we re-extract from the decoy signals in the raw data, we can, then the decoys will have either, because these are, the fragments have been swapped, this is not the real molecule, then we will see cases where the peak width will be deviated, or the retention time, or there will actually be noisy signals, and then the similarity with the reference spectra, it will have a low score, low spectral similarity. As I mentioned, we use Skyline to then do the extraction of the signals, the target extraction, after we generate the targets and decoys, and also to extract the signals from the library with our metabolite library. Then we perform the training. Our code, it just takes this list of metrics where we know what are the targets and decoys, and then we can train this support vector machine to distinguish between the metrics that are from a good peak group, or good signal quality peak group, or a bad one, or the decoy. And as we can see here, these are all the peak metrics that we use to train our algorithm, our machine learning model. Each of these individual scores, as we can see, it doesn't do a great job in separating, distinguishing between the targets and decoys. But when we combine it into the PeakDecoder score, that it's a combination of all these different metrics together, we can see that there is a clear distinction. The algorithm can learn how to distinguish true coelution comobility from the ones that are decoys that are low quality. The last step is the inference. We load this model that is already trained, and then we score the signals from the extracting it using our metabolite library as a seeds to do the extraction of the peak of the metrics. We build a library of 64 metabolites, which are relevant for the study of these different microorganisms, and we were able to annotate features from 26 more than 2,600 features and across 100 existing microbial samples. And even for the metabolites that we could not detect with fragments, because we have this library with the retention time and the collision cross-section, we can increase the confidence of that metabolite annotation. For the validation, we investigated and visually inspected how the signals look from our standard comparing the microbial sample. And we can see in this case, this example molecule, that the fragmentation pattern, the abundances are similar, and the chromatography peaks the elute, and the mobilities also is where we expect it to be for that molecule. And we did a manual validation and compare our results against, if we use MSDI alone, so we could see that PeakDecoder could increase significantly the number of annotations that we were able to do. And we also were able to decrease the number of false positives compared with Skyline, because we include false discovery rate analysis in our workflow. We performed also dilution experiments for selected metabolites. We could see that compared with selected reaction monitoring, we were able to quantify it across 4 orders of magnitudes, and with linear, with the expected linear abundances. And then we can also see here an example on the right of the chromatograms with SRM compared to our multidimensional workflow. And one of the reasons we can achieve more higher dynamic range, it's because in the DIA data, in this data, we can combine the abundance of the intact precursor plus the fragments, and we can do this post-acquisition. But in SRM, we are only measuring the abundance based on the fragments that are selected. We also compare metabolites in common. We measure also this with SRM and the real samples, the microbial samples by SRM and by GCMS, and we observed that there were similar trends in the relative abundances across the signals for the metabolites that were in common. So PeakDecoder was developed in the context of the IR Biofoundry Consortium of National Laboratories for Synthetic Biology. For this, we implemented our workflow and evaluated to study these microorganisms. We developed the chromatography methods and the ion mobility instrumentation parameters done by Natalie Munoz, Yuchan Gao, and Danny Orton. In this example, I would like to show you some results related to the Pseudomonas butida organism. Basically, here we compare the wild type strain with 3 other modified strains of this organism. And in this example, we see one of the comparisons between the wild type, and we could basically confirm that the modifications done to the organism were actually activating and producing the metabolites for what they were designed for. And in this example, we can see the metabolites are the dots, and the arrows represent the enzymes that we also measure and compare with the proteomics results. In this image, this figure, we compare how, if we didn't have, like, what's the power, what is the resolving power or the discrimination power that each of the dimension has, and what is the meaning of using all the dimensions together. And we can see here that compared to the number of matches, if we use one or all the the dimensions together, we can see that if we use only the mass for specifically more for the smaller metabolites, the number of features that we can match from the complex sample increase. So it means the ambiguity increase. But when we combine all the mass, the retention time, the collision cross-section, it really increases our confidence and the discrimination power. And in this way, because we can distinguish isomers in the samples and increase our confidence in the annotation and also in the quantitation. In the second project, we used also PeakDecoder in this workflow to analyze the fungal garden ecosystem. Basically, here we are interested in understanding the biomass, the process, the molecular process for biomass deconstruction, and here we combine multiple analytical techniques and a platform called MIPI, Metabolite-Informed Proteome Imaging. Basically, we perform imaging in different sections of this biomass deconstruction process to study more accurately, because if we do analysis by traditional LCMS, just in bulk, it means processing all together, we are diluting important signals that we are not able to distinguish. So in this integrated platform, we can focus our analysis in specific regions, and then we use these different platforms to confirm the identity of the metabolites that we were detecting and the proteins. And in this example, I would like to show you that using PeakDecoder in our multidimensional analysis, we were able to actually confirm more of the metabolite identity compared to the other platforms. Finally, I would like to talk about our current work developing PeakDecoder 2. PeakDecoder 2, it's being developed based on deep learning. What is the difference between machine learning and deep learning? The main difference is that deep learning, it can detect more complex patterns in the data, and we don't need to perform this feature extraction step. It means in the first version of PeakDecoder, we need to specify, we needed to design, okay, we will use the retention time, peak width, the mobility difference, the fragmentation pattern. So we are encoding all these features to tell the model what are the important metrics to use. But using deep learning, the model will learn how to extract those by itself. And as more data we have, the model is able to discriminate more and capture that complexity, model the complexity. But then deep learning has been already done great progress in the mass spectrometry field. In general, more for predicting properties. But our focus now is, can we use it also for pattern recognition in the 4-dimensional data directly? So beyond the prediction of individual properties. And we are using for this transfer learning. So you can imagine instead of detecting pedestrians, if we have an algorithm, a model that detects pedestrian, we can say, okay, now we want to detect peaks. We can take a model that is already able to recognize pedestrians in pictures and then tune it. It means we will now, the model already knows how to distinguish shapes and patterns in the figures, but now we will tune it to specifically recognize peaks that coelude and have a good comobility. The first limitation that we had in this development was the access to the raw data. For this purpose, we designed the EMSA data conversion tool. This is a tool to convert the raw data from the multidimensional analysis into a format that is much easier to work with and develop the tools. Is basically based on the HDFI binary open format, and it's the EMSA has a self-contained conversion software that can be executed without any installation of the dependencies, not even Python. So it's very easy to convert the data, and then we can develop our code software with Python or R and any other computational language. Here we have an example on the right, where we have a signals, visualization of raw signals for LC with slim ion mobility, another type of ion mobility, developed here in our lab. And we can see we could see the signals. We can basically represent it in the same way as the vendor software, but all based in Python code. So to do this utilization of the deep learning algorithms for image analysis, recognition, object detection in images, we need to do a visual, we need to create a representation of our of our raw data into suitable format. So on the left, we have the traditional representation based on chromatograms or heat maps of these 4 dimensions. So what we are doing is extracting these 2-dimensional ion signals. For a given mass, we can represent the retention time and the mobility in a single image. And we can do that for the fragments and the precursors. And basically, we're in the areas where we see that they align, it means that the molecule is present in the sample. This is an example for proteomics, another data set that we are evaluating. And we already can see that the model will be able to extract the signals and align even in very complex backgrounds when the signal is noisy. And we have in this example a light and heavy peptide. What we are doing is basically aligning these 3 signals, 3 images into a single image, and where we represent the 3 colors, and where basically the white areas are the ones that will tell us if they align well and where in the image exactly. In this example, we in our preliminary results, we see that the model is able to locate where in retention time and mobility these signals align. So that will tell us our actual coordinates in each of the samples that we can compare also against our library to measure the error. And in this case, we have an example of a confidence 0.85. In our current workflow, we are using the PNNL preprocessor tool and the EMSA then to improve the signal or the quality of the raw signals. Then doing the raw data conversion to EMSA. We are training our model based on curated data examples that we are extracting. And then we are using that trained model. We are working on creating a software executable that can do the inference, and we can use it to analyze at scale new samples. This brings me now to the conclusions of my talk. The combination of retention time and collision cross sections with comobility and coelution and comobility significantly increase the coverage and confidence in overall the analyte identification. Our complete analytical workflow, including the PeakDecoder, it's applicable also to other metabolomics research areas beyond synthetic biology. And by learning the coelution and comobility patterns from the raw data itself, without relying on fragmentation rules, PeakDecoder enable the annotation and error rate estimation or all other omics too. And we are currently developing PeakDecoder 2 based on deep learning to derive new molecular knowledge in an automated, faster, and more accurate fashion. To conclude, I would like to acknowledge all the amazing scientists and researchers that have worked in these projects and these have been involved in the results that I presented today. Specifically, especially to Kristen Vernon-Johnson, that is the PI in these projects, and the PeakDecoder 2 team that is now working, developing this new version. And now I'll be happy to answer any questions. Thank you.